This is now chapter 3. We have already looked at the molecular basis of gene expression, and we've looked at the process of meiosis, where cells are prepared for gametes, which then go on to the next generation. So this is the genetics behind both of those processes, first discovered by Mendel, of course, back in the 1800s. This is a picture of Mendel and his friends. Can you tell which one is his? It's to my right there. That's Mendel. And here's a close-up of Mendel. He was born in 1822, which is now the Czech Republic. He taught physics and natural history. He had a very logical mind, which is a very good scientific basis. At age 21, he entered a monastery in Brno and finished studies there some 30 years later, and then studied natural history in the University of Vienna. Not that this is a memorization course for genetics, but it gives you an idea of the kind of background and education that Mendel had. And in 1854 he returned to Brno to teach physics, but he was more interested in a number of things, including why male and female gametes were of different size, why the pollen of a plant was a little smaller than the ovum of a plant. And so he did some work in a garden in the monastery, and here's the picture of the garden today. And he designed some very well thought out experiments to address this question of unequal size of gametes. So these were the traits that Mendel looked at in his peas. As you can see, there are seeds, different shapes and colours, and some pod shapes and colours, flowers where they are, and stem lengths. And these are the dominant and recessive phenotypes for those particular traits. And here's a cartoon of the same kind of thing, maybe a little better. We can see that we have round and wrinkled seeds, different colour seeds, different colour flowers, and so on and so on. So he used these 7 traits to study the inheritance patterns of these traits from one generation to the next. So let's look at one of the crosses that Mendel performed. In this case it is seed shape, whether it's smooth or wrinkled, and here is the smooth seed, and here is the wrinkled seed right here. What he did is he used pure breeding parents. These guys were pure breeding. Now what this meant is that if you took these smooth seed plants and self-crossed them, they would all produce smooth seeds. Ditto with the wrinkled seeds. If you self-crossed these plants, all their seeds would be wrinkled. So these were pure breeding genetically parent plants. So when you did a cross between the smooth seed plant and the wrinkled seed plant and produced the next generation, which is called the F1 generation, all the plants were smooth. Which was kind of strange, isn't it? I mean, where did the wrinkledness go to? Well, so this next step was to self-fertilize these F1 plants. And when you self-fertilize them, you can do this with plants. You just cross the pollen with its same flower, and that's called self-fertilization. And he produced nearly, well, just over 7,000 new seeds, and about 5,500 of those were smooth, and about 1,800 or so were wrinkled. And if you do the math on this, it works out to be about a 3-to-1 ratio. And so he did this again and again with all those different traits from the previous slide. And here are some of the results. And you can see here that across round and wrinkled seeds, yellow and green seeds, purple and white petals, and so on, here are the numbers, and here's the ratio. About 3-to-1 in every case. So what did this mean? Well, it meant the factors, or genes, as we now know they are, but he didn't know them, can be expressed in either a dominant or a recessive form. Of course, we now know that this is the dominant is the smooth, and the recessive is the wrinkled. Parents can pass on only one of their 2 copies of a gene onto the next generation. A version of a gene is called an allele, and the parent has 2 of every kind, and so you can pass on 1. So you have pass on one allele for smoothness, and pass on one allele for color of the seed, and so on and so on. The F1 plants then carry 2 factors for each trait, or 2 alleles for each trait, one from the male parent and one from the female parent. So from this, Mendel produced not what he called the first law, but we call Mendel's first law, which is the principle of segregation. And it says that the pairs of genes separate from each other during gamete formation. In other words, alleles separate during gamete formation. And on this diagram we can see here are two alleles. These are two dominant S alleles, and they separate during meiosis. Same thing for the wrinkled. Here are two recessive alleles. These are lowercase letters, and they separate during gamete formation. And when you go on to make the next generation, you take one of these and you take one of these, randomly so, combine them, and this is the genotype of the next generation. So we have one allele from one parent, one allele from the other parent in a random format. Combine them together, and this is the genotype combination for the next generation. So a little bit of few definitions. An allele then is one possible alternative form of a gene, usually expressed as a recessive and a dominant version. Recessive is lowercase, dominant is uppercase. And homozygous, whether you have two the same, it could be dominant or recessive. And heterozygous if you have two different alleles, as in here. So creating the next generation, creating the F2 generation, we take the F1 generation that we know is heterozygous, and we're going to make the F2 generation. And here's that 3:1 ratio. Here's a smooth, 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 and wrinkled. And so we have 1 wrinkled to 3 smooth. So how does this happen? Well, let's start at the beginning up here. From the smooth generation, we can produce 2 different alleles, big S and big S. And from the wrinkled parent, we can produce 2 alleles, little s and little s. And if we put this together in what we call a punnet square, we put these alleles on one side, these alleles on the other side, and just add them together. So in every case here we have a big S and a little s. And this gives the F1 genotype. And as you can see, they're all big S, little s, or heterozygous. And we can do the same thing again with the F1 generation. What we'll do is we'll take the genotypes for the F1, put those alleles alongside, scrap that, put those alleles alongside a punnet square, ignore that, and if you add those together we have big S, big S, big S, little s, big S, little s, and 2 lowercase s's here. And so we find that these 3, these are dominant, and this one here is recessive, which means these 3 are smooth, and this one here is the recessive. And so you can see here we have a 3:1 ratio. Even though 2 of these dominant traits are in fact heterozygous, we're still 3 dominants to 1 recessive. Well, once Mendel had figured out his initial findings, he then went on to cross 2 traits. And what we do now is we cross 2 separate phenotypes, such as smooth and wrinkled and yellow and green, and so on and so on. And what Mendel showed was that members of one gene pair, in other words, for color, separate or segregate. That's what separate means and segregate means. One gene pair segregates independently of the other gene pair. In other words, the color of the seed has nothing to do with the shape of the seed. And this is called independent assortment. And let's have a look at that and see how that works out. So here's crosses with 2 traits. Same thing again, we have a parental cross, and these again, of course, are pure breeding. So a smooth yellow seeded plant, if self-crossed, will produce all smooth yellow seeds. And a wrinkled green plant, seeded plant, will, when self-crossed, will always produce wrinkled green seeds. So this is the parental cross. We cross this cross this one with the other, and we get the F1 generation. And what we find here is that the entire F1 is smooth and yellow. So now if we take the F1 generation and cross it with itself, which is here, to make the F2 generation, the next generation, we find that there are a whole combination of different traits arising. Smooth yellow, smooth green, wrinkled yellow, and wrinkled green seeds, all possible combinations of the original parental cross. Let's work this out in a Punnett square to see how this works. First of all, we need to work out the alleles for each of these parents, and we know they're pure breeding. So this one is going to be big S, big Y for smooth and yellow. And this one over here is little s, little y, because we know it's wrinkled and green, and we know they're pure breeding. So the alleles coming from this parent are going to be big S and big Y. That's the only possible combination. It's going to be one of these two and one of these two, so this is the only thing it can be. Same over here, it's going to be little s and little y. So if we combine those together, we don't really need a Punnett square for this. We combine those together, we know that it's big S, big Y, little y.